Hello, welcome again to this new course of plant physiology and metabolism. So, next essential mineral element is sulfur. You see that sulfur stands as one of the essential nutrient for the plant, which is crucial for the growth and development of all crops. It is generally sulfur is found in amino acids like cysteine and methionine, and is you see that constituent of several coenzymes and vitamins such as coenzyme A, S-adenosyl methionine, biotin, vitamin B1 and pantothenic acid, which are very essential for the various metabolic processes. So, like other essential nutrients, sulfur also serves pivotal function in plant, which we will discuss in our coming part of the lecture. Now, in what form this sulfur is taken up by the plant? So, the main source of sulfur is basically sulfate, which is taken up by the roots. However, higher plants also absorb and utilize atmospheric sulfur dioxide through their aerial parts. Within the physiological pH range, the root cells absorb divalent sulfate anion, which is then transported throughout the xylem and phloem. So, this transportation process involves transmembrane steps facilitated by a family of sulfate transporters. So, the assimilate of sulfur shares many similarities with nitrate assimilation. For example, reduction is essential for incorporating sulfur into amino acids, protein and coenzymes. Generally, in green leaves, you see that ferredoxin acts as a reducing agent for sulfate. Unlike nitrate, nitrogen sulfate can also, unlike nitrogen, sulfate can also be utilized without the reduction and integrated into the essential organic structures such as sulfolipids in membranes or polysaccharides like agar. Additionally, unlike nitrogen, reduced sulfur can be reoxidized in plants. This oxidation reaction converts reduced sulfur of cysteine to sulfate, which is considered the safest storage form of sulfur in the plants. Now, just this is a brief overview of what sulfur imparts, that what is the important role of sulfur, just we have basically discussed. Now, how sulfur is taken up, its physiological role and specific deficiency symptoms, and then finally toxicity symptoms imparted by sulfur. So, you see that usually plants absorb sulfur in the form of sulfate ion, and plants could also get sulfur from the atmosphere, and generally sulfates are reduced in plants to sulfohydryl group, and then its assimilation of sulfate basically requires the reduction of sulfate to cysteine. Now, what are the important metabolic functions of sulfur? You see that sulfur serves as a very crucial component, structural components of amino acids, and you see that amino acids containing sulfur are particularly vital for protein structure, enzyme function and various metabolic process in living organisms. So, one of the most notable sulfur containing amino acids, you might be knowing that it is cysteine, and cysteine is very unique among amino acids due to its presence of thiol group, that is SH group, which allows it to form disulfide bond with other cysteine residues in proteins. So, this is the unique feature. So, these sulfide bonds contribute significantly in the stability and structure of many proteins, particularly those secreted outside the cell or found in extracellular matrices. Then another sulfur containing amino acid is methionine, which serves as a initiating amino acid in protein synthesis and acts as a precursor for the synthesis of S-adenosyl methionine, which serves as a very crucial methyl donor in numerous biochemical reactions. Basically, this adenosyl methionine is involved in DNA methylation, histone modification, which serves as a neurotransmitter synthesis, and various metabolic pathways. Now, how synthesis of sulfur containing compounds are achieved? You see that sulfur plays a very important role in the formation of sulfur containing compounds, which are crucial for various biochemical processes across the living organisms. One prominent group of sulfur containing compounds is thiols. So, these thiols are organic compounds characterized by the presence of a sulfohydryl functional group you see in the figure. And then these thiols basically play a very essential role in protein structure, as cysteine residues could form disulfide bonds with other cysteine residues, contributing to the stabilization and folding of the proteins. So, basically disulfide bonds are crucial for maintaining the tertiary and quaternary structure of proteins, and they often determine the stability of proteins and their function. Another significant sulfur containing compound is glutathione, which is a tripeptide composed of cysteine, glycine and glutamate. So, glutathione serves as a very important, or you can say potent antioxidant, protecting cells from oxidative damage by scavenging the reactive oxygen species and free radicals. This also participate in detoxification process by conjugating with toxic compound, facilitating their removal from the cell. Thioredoxin you see are very small proteins that play key roles in maintaining cellular redox balance by catalyzing the reduction of disulfide bonds in the target proteins. These proteins contain conserved active site sequence with a redox active disulfide bond. The sulfur atom you see that in the disulfide bonds are essential for ability of thioredoxin to shuttle reducing equivalents between proteins participating in processes such as DNA synthesis, protein folding and the scavenging of reactive oxygen species, that is ROS. So, the sulfur atoms present in the thioredoxin are derived from sulfur containing amino acids such as cysteine, further emphasizing the importance of sulfur in maintaining the functional integrity of these proteins. Now, phytochelatins you see that these are the small peptides synthesized in plants in response to heavy metal stress. So, plant could face various sort of different heavy metal stress, particularly exposure to like cadmium, lead and arsenic. So, these peptide chelates, these peptides basically chelate heavy metals ions through the thiol group of cysteine residues, forming stable compounds that are sequestered within the vacuoles and thus preventing the metals from causing damage to cellular structure and biomolecules. In addition to this, you see that sulfur is also essential for the synthesis of phytochelatins, as you see that peptides contain multiple cysteine residues that provide the binding site for heavy metals. The synthesis of basically phytochelatins involves the sequential addition of cysteine residues to form a polymer with the sulfur atoms in the cysteine residues playing a central role in metal binding and detoxification. Sulfur is generally also present in various coenzymes and cofactors that participate in enzymatic reactions. For example, iron sulfur clusters are the prosthetic groups found in many enzymes involved in electron transfer reactions, such as those found in electron transport chain of mitochondria and photosynthesis in plants. In addition to this, sulfur containing compounds play a role in signaling pathways, you see, very important, and regulating mechanism within the cells. For example, hydrogen sulfide has emerged as an important signaling molecule involved in elevating abiotic stresses in plants. Many molecules synthesized by the plants in response to abiotic and biotic stresses contain sulfur. This includes phytochelatins, thyrodoxin, alins, glucosinolates, etc. So you see that the alins are found in onion and garlic, while glucosinolates, they are found in the members of family Brassicaceae, and they are responsible for specific flavor and smell. In some plants, elemental sulfur is basically deposited, which functions as a potent fungicide. Now, generally reduced sulfur serves as a structural component in various coenzymes and in prosthetic groups found in biological systems. You see, these includes ferrodoxin, biotin known as vitamin H, and thiamine pyrophosphate, commonly referred to as vitamin B1. Additionally, the sulfohydryl group within the numerous enzymes and coenzymes such as urease and sulfotransferases and coenzyme A play crucial roles as functional group in enzyme mediated reactions. So you see that within the glycolytic pathway, specific enzymatic process, they are facilitated by sulfur containing coenzymes. For example, the decarboxylation of pyruvate and the subsequent formation of acetyl coenzyme A are basically catalyzed by multifunctional enzyme complex involving 3 sulfur containing coenzymes, and that is thiamine pyrophosphate, sulfohydryl disulfideox system of lipoic acid, and the sulfohydryl group of coenzyme A. So following this enzymatic process, the resulting acetyl group of coenzyme A can be further esterified, you see in the figure, into the tricarboxylic acid cycle or TCA cycle or to fatty acid synthesis pathway. So in addition to this, the synthesis of long chain fatty acids further necessitate the coupling of carbon units. This process basically involves the transient carboxylation, which is generally mediated by another sulfur containing coenzymes, biotin. So the activity of biotin is in this regard is facilitated by the activation of manganese. So the incorporation of sulfur into various coenzymes and prosthetic groups is basically pivotal for numerous metabolic pathways, ensuring the efficient functioning of biochemical processes essential for cellular metabolism and energy production in living organisms. Now, once plant faces any sort of deficiency symptom, plant reflect it in various parts of the plant. So you see that whenever the sulfur concentration in growing plants drops below a certain critical level, generally plants reflect visual symptoms of sulfur deficiency which start appearing in the plant. Now question arises, where this deficiency symptom appears in case of sulfur. So sulfur is mobile element or immobile, question arises. So during the classification I had discussed that sulfur is immobile element. So the appearance of sulfur deficiency generally likely to appear in where, whether it is in the younger part or older part. So because sulfur is immobile, so its initial deficiency symptom will appear in the younger part. So sulfur also induces numerous deficiency symptoms and that closely resembles with nitrogen deficiency. It is very interesting to note here that the deficiency of sulfur closely resembles with deficiency of nitrogen in plants such as chlorosis, growth stunting and anthocyanin accumulation. So this resemblance is due to because both sulfur and nitrogen are components of proteins. But you see that there is a very contrasting point at this juncture. This chlorosis resulting from sulfur deficiency arise first in the both young and then extend up to mature leaves. And this is contrasting with the nitrogen deficiency, which primarily affects older leaves. So this distinction arises because sulfur, unlike nitrogen, is not readily remobilized to younger leaves across most species. So generally plant deficient in sulfur, they are small and spindly with short and slender stalks, and their growth is retarded, and defoliation in tea plants, nodulation in legumes may be poor, and nitrogen fixation is reduced. Fruit often do not mature fully and remain light green in color, and generally forage contain an undesirably wide nitrogen sulfur ratio and thus have lower nutritive value. So at this juncture, all of you must be aware that both nitrogen and sulfur deficiency induces chlorosis, but their localization in different parts of the plant is totally different. One is localized in younger part at the initial stage, another one is localized in the older part. So you must be aware that visual deficiency symptom of sulfur and nitrogen are same, but their localization in plant is different. So you should note this very important point. Now moving on, next is magnesium. Magnesium is also a very important essential element required by the plant. So we will discuss that in which form this potassium is taken up, this magnesium is taken up by the plant, and how it affects the growth and development and overall health of the plant. So basically magnesium is considered one of the important essential macronutrient for the plant, and they are required by the plant in generally large quantities for proper growth and functioning. And you see that like other essential mineral elements, magnesium is also absorbed by the plant roots, but their form is different, primarily as a divalent cation. And once magnesium is absorbed, it basically travels to the xylem vessels, where it may be transported in either free form or bound to other molecules in chelated form. And generally magnesium deficiency in soil is not very common, but acidic and sandy soil can pose challenge to its solubility to plants, often due to pH related effects. In soil solution, generally the concentration of magnesium ranges from approximately 0.1 to 8.5 millimole. However, within the cytosol of the plant cell, the concentration of magnesium is notably very low, averaging around 0.4 millimole. So the uptake of magnesium into the plant root is generally facilitated by specialized plasma membrane magnesium channels, predominantly belonging to MSR2 family. So you should remember the type of channel involved in uptake of magnesium at this juncture. This is very crucial. These channels allows for the selective passage of magnesium ions into the root cells. Once magnesium reaches inside the root cells, it navigates across the tonoplast, the membrane surrounding the vacuole. So this movement across the tonoplast, you see, is facilitated by the simple transport proteins, namely MHX magnesium proton antiporter and the TPC1 magnesium permeable cation channels, ensuring magnesium ions reach into their destination within the plant cell. The absorption of magnesium, you see that can be significantly inhibited by other positively charged ions such as potassium, ammonium, calcium, manganese, as well as by low pH levels caused by excess protons. Therefore, competition from these ions could lead to widespread magnesium deficiency in the plants. Now, once potassium reaches to a specific destination, now its role is start, we could say its metabolic roles. So, like other essential mineral elements, magnesium also serves as a pivotal component of the chlorophyll, which is predominantly found within the porphyrin moiety, which contributes to green pigments found in the plants. In addition to its role in chlorophyll, magnesium acts as an essential enzyme cofactor, facilitating the production of adenosine triphosphate, that is ATP, which serves as the primary energy currency within the cells. We have gone into the details. In addition to this, this is very important to note here that magnesium is crucial for the binding of ribosome subunits, thereby aiding the synthesis of proteins. So, basically it helps binding of the different subunits of the ribosome. So, magnesium also serves as an activator and plays a crucial role in the activation of enzymes involved in dark reaction of photosynthesis. Most importantly, enzymes such as ribulose bisphosphate carboxylate, that is BISCO, and phosphoenol pyruvate carboxylate, that is PEP carboxylase, rely on the magnesium ion for their proper functioning. Beyond its role in photosynthesis, magnesium is also very much indispensable for various enzymes involved in cellular respiration and the biosynthesis of nucleic acids. So, involvement of basically magnesium in these fundamental metabolic processes underscores the significance of this essential mineral element and various processes occurring in the plant. Now, role of magnesium in the formation of chlorophyll. You see that chlorophyll is synthesized and heme follows basically a shared pathway until the formation of protoporphyrin 9. You see, at this juncture it is very crucial that basically in the initial stage of chlorophyll biosynthesis, the insertion of magnesium into the porphyrin structure is basically facilitated by magnesium chelatase. The activation of, you see, in the next activation of this enzyme also require ATP, which further require additional magnesium. And during the chlorophyll breakdown, the release of magnesium occurs through two sequential steps, that are chlorophyllase hydrolyzes chlorophyll into chlorophyllide and phytol, and magnesium dechelatase liberates magnesium along with pheophytin. So, as I mentioned earlier, that magnesium impart very crucial role as a linking element for binding together the ribosomal units, which is a very vital step in the synthesis of protein. So, in conditions of you see that magnesium deficiency, or when high level of potassium are present, the subunits separate, halting the process of protein synthesis. Additionally, magnesium plays a pivotal role in stabilizing the specific structure of nucleic acids, essential for their synthesis and functioning, as well as for the activities of nucleic acid polymerase and nuclease. Now, what is the role of magnesium in the activation of enzyme? You see that magnesium plays a very crucial role in the activation of enzymes in plant, including rubisco, glutathione synthase and polyphenol phosphoenol pyruvate carboxylase. You see that how magnesium contributes to the activation of these enzymes. First one is rubisco. Rubisco, we have gone through in detail. Rubisco is the key enzyme involved in Kelvin cycle, where it catalyzes the carboxylation of RUBP to form two molecules of 1,3-phosphoglycerate, which is a very crucial step in carbon fixation during photosynthesis. In addition to this, magnesium is also required for the activity of the enzyme rubisco, and basically rubisco is inactive in the dark and gets activated in the light. So, rubisco is activated by binding of carbon dioxide and magnesium ions to the lysine residue near the active site, which brings about conformational change and stability of the active form of the enzyme. Now, another enzyme is glutathione synthetase, which is an enzyme involved in the synthesis of glutathione, which serves as a potent antioxidant and crucial molecule for the cellular redox homeostasis. So, basically magnesium serves as a cofactor for glutathione synthase. Now, role of magnesium in activation of phosphoenol pyruvate carboxylase. Basically, phosphoenol pyruvate carboxylase is involved in the carbon fixation during the photosynthesis, particularly in C4 plants and CAM plants. So, in the presence of magnesium, the substrate phosphoenol pyruvate is generally bound to greater quantities and more tightly. So, here also magnesium is imparting very significant role. Then another enzyme is fructose 1,6 bisphosphate, which is another enzyme involved which requires magnesium for proper functioning and high pH generally. Fructose 1,6 bisphosphate generally regulates assimilate partitioning between starch synthesis and export of triose phosphate in the chloroplast. So, this is very important role of magnesium in the activation of enzyme activity. Now, like other essential mineral elements, magnesium also imparts a specific deficiency symptom. You see in this slide on the left side, you see that one leaf is deficient, another leaf is having significant amount of adequate amount of magnesium. So, generally magnesium also induces a specific deficiency symptoms, and the specific deficiency symptoms that is induced by magnesium is intraveinal chlorosis, which is characterized by yellowing between leaf veins, and this is very significant to observe this sort of deficiency. And magnesium deficiency also leads to reduced rate of photosynthesis per leaf area or affect chlorophyll photosynthesis, resulting in carbohydrate accumulation. Generally, during the deficiency of magnesium, root growth is hindered in plants, and inadequate magnesium availability disrupts the export of carbohydrates from sucrose to zinc tissues, leading to decrease in starch concentration in storage tissues. So, what is the mobility of magnesium? Yes. So, magnesium is basically a mobile element that just we have gone through it. So, older leaves are primarily affected, displaying the accumulation of anthocyanin, and premature leaf abscission. So you see that if plants face any sort of deficiency of magnesium, interveinal chlorosis first will appear in the older leaves because of the redistribution to younger parts, and then if deficiency of magnesium continues for longer duration, it is going to extend up to the growing region of the plant. So in this way, the initial deficiency symptom of magnesium is very easy to detect, that is characterized by interveinal chlorosis. So this is all about the role of magnesium specific deficiency symptom. In our coming lectures, we will discuss another essential mineral elements. Thank you very much.